In the previous video, we discussed about agammaglobulinemia. If you want to watch that video first, the link is in the description. Now in this video, we are going to discuss about the pre-BCR signaling, its role in B cell development. Like we know, it's a crucial checkpoint in early B cell development that ensures only properly rearranged B cells move forward. And we will be also looking for the diseases that are related to this pre-BCR signaling, like we have agammaglobulinemia, which occurs due to absence of BTK protein or defected protein, which is a part of this signaling pathway. Now let's explore the pre-BCR signaling pathway in detail. Here we see in this diagram we have the plasma membrane of pre-B cell. On this plasma membrane we have Pre-B Cell Receptor complex embedded in the membrane. This complex is made up of mu heavy chain paired with surrogate light chains VPREB and gamma 5. And the important signaling molecule is CD79A and CD79B. Both these protein molecules of CD79 contains ITAM motifs, which play a central role in transmitting the signal inside the cell. Also present in the membrane is PIP2, phosphatidylindole 4,5-bisphosphate, shown in the diagram, which will be used later in this pathway for second messenger generation. Now let's look at how the signal is initiated. At this early stage, there is no external antigen driving receptor engagement. Instead, the pre-BCR complexes tend to spontaneously cluster on the surface of the cell, a process known as self-aggregation or tonic signaling via cross-linking, shown in the animation. As a result, a SRC family tyrosine kinase called LYN is recruited to the receptor complex as shown in the diagram. LYN is membrane associated and becomes activated upon receptor clustering. Once active, LYN phosphorylates the ITAM domains on CD79A and CD79B. This phosphorylation creates docking site for the next molecule in the cascade, that's SYK, a cytoplasmic tyrosine kinase. So in the next step, we see SYK binds to the phosphorylated ITAMs through its SH2 domains and becomes activated, either through autophosphorylation or further phosphorylation by LYN. Moving on, SYN then phosphorylates downstream adapter molecules, specifically CD19 and BCAP, that's B cell adapter for PI3K molecule. These phosphorylated molecules or adapters now act as recruitment platforms for PI3K. At this point, PI3K complex is getting recruited to the membrane. We see it gets recruited in units. First, we get the recruitment of P85 regulatory subunit, followed by the P110 catalytic subunit of PI3K complex. So ultimately, we get the complete PI3K complex. It gets activated. Once activated, PI3K catalyzes the conversion of membrane-bound PIP2 into PIP3 as shown in the animation. Now that PIP3 is generated, it acts as a docking site for BTK protein. BTK has pH domain that specifically binds to PIP3, anchoring BTK to the plasma membrane. Once at the membrane, BTK is phosphorylated at tyrosine 551, typically by the LYN or SYK, and then undergoes autophosphorylation at tyrosine 223, becomes fully active here. With BTK active, the signal continues. BTK now phosphorylates and activates PLC gamma 2, that's phospholipase C gamma 2, another key player in this pathway. In the following step, PLC gamma 2 hydrolyzes PIP2 still available in the parts of membrane into two critical second messengers, IP3 and DAG. It must be noted here, PIP2, if acted by PI3 kinase enzyme, it gets converted into PIP3, or if PIP2 is acted by PLC enzyme, it gets converted into DAG and IP3, like in this case. Getting back to IP3 and DAG, we see IP3 diffuses into the cytoplasm and triggers calcium release from the endoplasmic reticulum, whereas DAG, which stays in the membrane and activates PKC, that's protein kinase C. These second messengers now activate key transcription factors inside the nucleus. IP3 mediated calcium release activates calcineurin, which in turn activates NF80, nuclear factor of activated T cells. Second is DAG. It activates PKC, which leads to activation of NF-κB pathway. Simultaneously, other branches like the RAS-MAPK pathway gets triggered, resulting in AP1 activation. All these transcription factors, NF-AT, NF-κB, and AP1 drive gene expression programs essential for pre-B cell survival, proliferation, and further development into immature B cells. Here we also get the regulation of several processes like allelic exclusion, VDJ recombination checkpoints, and cell survival. And finally, it's important to note defects in this pathway, especially mutations in BTK, can severely block B cell development, leading to X-linked agammaglobulinemia, a condition marked by absence of mature B cells and low antibody levels. So to sum up, the pre-BCR signaling pathway is tightly regulated, multi-step cascade that begins with receptor clustering and ends in the activation of transcription factors, ensuring that only functional B cells move forward in their development. So this is all about pre-BCR signaling. I hope you like the video. If you like it, give it a thumbs up. You can support my work on Patreon or YouTube and make sure to subscribe to the channel. Thanks.